Hello. In this lecture video, I'm going to spend a few minutes discussing the nutritional requirements of microorganisms. There are a lot of different types of media that we can use in order to grow microbes. So far in the lab, we have just used nutrient agar or nutrient broth. And this is kind of our basic all-purpose media that we use for growing a wide variety of organisms. But we can use very specific media, which we'll do as we continue on through our microbiology course. So all-purpose media would be like the nutrient broth that we've been using. It can support growth of a large variety of organisms. Another type of all-purpose media would be tryptic soy agar, which we'll use a little bit later in the semester. Specialized media can be used to identify bacteria or to be infused with dyes or antibiotics or other materials. One specific type of specialized media is referred to as enriched media. And this type of media includes very specific growth factors or nutrients or vitamins that promote the growth of what we refer to as fastidious organisms. Fastidious organisms are those that are essentially picky eaters. They require very defined, very specific nutrients and other growth factors. So we have to use special agar to grow those. With selective media, this type of medium inhibits the growth of certain organisms and supports the growth of the organisms that we're trying to grow. One example of this is Mcconkey Auger, and we'll use this later on in the semester when we study our enteric bacteria, specifically the family Enterobacteriaceae. Mcconkey Auger contains bile salts and crystal violet, and this interferes with the growth of a lot of gram-positive organisms, and it allows us to really just grow gram-negatives. And specifically our enterics, the ones that reside in the intestine. They're adapted to grow in the presence of bile salts. So by adding the bile salts and the crystal violet, we're essentially selecting to grow these particular organisms. Another type of specific media is differential media. With differential media, we're going to get some type of color change, either in the colonies themselves that are growing on the media, or the color of the media itself will change. Mcconkey Auger is also an example of differential media. Mcconkey Auger allows us to observe fermentation of lactose. If we have organisms that are growing on the Mcconkey Auger that produce lactose, or that ferment lactose to produce acid, then it will turn the fermenters hot pink. So we'll see this bright, vibrant pink color. Another type of differential media that we'll use is mannitol salt agar. Mannitol salt agar has phenol red in it, and if we get acid fermenters growing on the mannitol salt agar, it turns yellow. So differential media allows us to really distinguish between different types of bacteria by looking at the color change. Now, another couple of definitions that you'll see here are chemically defined media and complex media. When we know the exact composition of a particular growth medium, that's referred to as a chemically defined medium. But in complex media, we don't know. We know what it contains, but not necessarily the precise chemical composition. Um, a lot of our, general or all-purpose media are examples of complex media. We know the basic ingredients, but we don't know exactly how much of each one. Now, here is an example. This is an example of a Mcconkey Auger plate. Okay. So, first of all, this is selective because we have, in this example, we've got a couple of different gram negatives growing on here. So it's selective for that. And then you see one set of the colonies is hot pink. And so that is the E. coli. E. coli ferments lactose, and as it ferments lactose, it produces acid, which allows the colony and the auger to change to this bright pink color. So that brings us kind of to the end of this section. This was a really short section that we went through. But we'll experiment with some of these as we go throughout the semester. Like I said, we'll use the Mcconkey auger, we'll use mannitol salt auger. We will look at growing streptococcus on blood auger. We use urea broth, phenol red broth. So we use a wide variety of media that can be differential or selective, or in some cases both.